Okay, welcome to this little video, which is just a very quick review of the cell and its organelles. Something that is covered in anatomy and it should be covered in a previous biology course. But since we're going to be talking about some of the organelles in a bit more detail, it's actually worthwhile just to have a think about what they do for the cell itself. Now, in the longer PowerPoint, you'll see that we talk about prokaryotes and eukaryotes, etc. But this is all based around the eukaryotic cell, which is what we as humans are made up of. But the bacteria, which actually in cell terms outnumber the human cells by not by much, about I think like 57% bacteria, 43%. Human cells was the last calculation that was published. They are prokaryotes, so they're just basically a cell membrane, a nuclear region without membrane, and a few organelles like ribosomes. However, for us, a little different. We have a much bigger cell. If you draw a bacterial cell next to this, it's going to look like this. And this nuclear region and a few ribosomes. So much bigger. And a lot more going on. Now, the first question you can ask yourself is, well, if the bacteria just, have kind of an... You could compare it, a bacterial cell and a human cell to something like a fifth wheel to a mansion house with, 14 bedrooms and 5 bathrooms or something of that nature. The eukaryotic cell, or the cells that make us up, are like the mansion. There's lots of different rooms with different functions. Whereas the bacterial cell is more like a fifth wheel, which is essentially something which is, or travel trailer, which is you're almost living in one room, pretty much. The older ones in particular. So, why bother with all of these rooms? Well, it comes down to specialization and chemistry. If for example, the mitochondrion here, which produces energy in the form of ATP. Wants to get its job done, it has to work in very different pHs. The pH in the intermembrane space there is about pH of 1 to pH of 2, whereas the pH in the cytoplasm is 7. So any of the chemistry that takes place in the cytoplasm, which is just the fluid and all of the organelles floating about in there, has to be able to work at pH of 7. But if you want something to work at pH of 2, then you have to create a membrane and put it in a specific room for it to be able to do that without it having any effects on the wider chemistry taking place in the cell. So eukaryotic cells can do lots and lots of different things, very specialized things, because they are compartmentalized. So what do we have? Well, we're going to talk about the cell membrane, which I've drawn here with the phospholipids and the integral proteins and the cholesterol holding everything together in the next talk. So we'll just say it's a double-layered membrane. Many of the organelles, such as the nucleus, mitochondria, etc., are all double-layered membranes as well. They've got some single-layered membranes like the Golgi apparatus, etc., and the rough and smooth endoplasmic reticulum. But they're all made up of phospholipids. Now, what do we have? So we have the nucleus at the center. Now in a eukaryotic cell, the nucleus is surrounded by a double membrane. That means it's protected, or well protected. And that's important because inside the nucleus, all this dotted stuff here is what we call chromatin. And chromatin, which I've written over here, is the chromosomes when the cell is not in division. Only when the cell goes into division can you see the little X-shaped chromosomes as we classically depict them. Normally we just see it like static on a television, but we know that the DNA is in there, and the DNA sequences of it code for proteins called genes, and we've got about 20 to 30,000 of them in our nucleus, and every nucleus contains the same amount. It's just that we only express a certain number of those to make a specialist cell. So this is well protected by this membrane. So when you get UV light coming in from outside, so you've got harmful radiation, it's first got to get through this barrier, then it's got to get through all of the organelles, So some of it get rejected there, some gets rejected there. I'm going to put UV, can't draw that very well. Radiation gets rejected there, some makes it through there and gets all the way to here but still gets rejected off. And very little of it gets into there. Whereas in a bacteria, it just has to get through there. So that same UV light, one might get knocked away, but lots of others go straight in. And that's why bacteria mutate so much quicker than eukaryotic cells' DNA does, because they're much more exposed. So this nuclear membrane, good for protection. It only breaks down when it's dividing, and that's when the cell becomes vulnerable. The dark region here is the nucleolus. I don't actually have that. Well, I have it written here, but I don't have it, specifically marked. The nucleolus creates the RNA, which is a type of nucleic acid, which has a number of various jobs, which we talk about in the later lectures. So there's our nucleus. Attached onto the side of the nucleus, we've got pores in the membrane to allow the products of protein synthesis to leave and enter the cytoplasm, or if that protein is going to function either here in the cell membrane or outside of the cell completely, then it moves into the rough endoplasmic reticulum. And it's rough because it's covered in these little red balls, and the red balls are ribosomes. Ribosomes are there because they carry out the second phase of protein synthesis called protein translation. And again, don't worry if that's not making a lot of sense, because in a week or two we're going to talk about that in detail. So if the protein is going to work in the cell membrane or outside, it doesn't enter the cytoplasm. Instead it enters this flat tubular network, all cell membrane, just flattened down and covered in ribosomes. And the ribosomes can take it and actually produce the protein inside these tubes, and then bud off in little vesicles. Got the wrong colour here, but I'll do it anyway. So it buds off, and these little vesicles carrying the protein can move over and then splurge them into there. And this is the Golgi apparatus. And the Golgi apparatus is kind of like, I always thought of it as a student as a travel agency, and they're less popular these days because of the internet, but they still have their place. The Golgi apparatus takes the protein and it adds things to the head of the protein, one end of the protein, and the other end. And these are essentially tickets to travel. They're special pieces of code that allow it to get through the membrane, and also will allow it to get in through the membrane of another cell, and potentially into the nucleus of another cell. For example, so they just give them tickets to ride, if you like, and so they make them ready for travel. So there's our rough endoplasmic reticulum and our Golgi apparatus. Up here we have our smooth endoplasmic reticulum, and endoplasmic just means really within the plasm or cytoplasm. Smooth means there's no ribosomes, therefore the protein synthesis is not part of what smooth endoplasmic reticulum does. Instead it works on fat synthesis and detoxification of species that are either produced in the cell or come into the cell and will cause damage. Alright, so the little red flecks in the cell, these are the free-floating ribosomes, which will produce proteins that act within the cytoplasm itself. These little orange balls are lysozymes. These carry, and you've already done a little bit of reading about these anyway, these carry enzymes. They've got specially coated on the inside and glycosylates, which help to prevent them. The enzymes are kept in an active form, but it helps to prevent them from being digested themselves. But they can wrap around things that are sent into the cell, or go in and release their enzymes onto special organelles and break them down, keep the stuff that's useful and get rid of the stuff which is damaging. Peroxisomes are probably floating around in there somewhere, and they basically, we've got some, you got a line for it and then nothing actually there, so let's do that. Peroxisomes, and they basically convert, what did I want to say, charged molecules, really, things that will cause damage. So hydrogen peroxide, for example, can be broken down to oxygen and water made safe. Mitochondria. This is something that we're going to look at in a couple of weeks, so all I'll say about it now is it takes various macromolecules, and it can basically produce the energy of the cell, and the energy of the cells, we'll find out in a week or so, is ATP, or adenosine triphosphate. And if that's familiar at all, adenosine, adenine, it comes from one of the bases of the nucleic acids with, instead of one phosphate, 3 phosphates attached to it. But we'll talk about that at a later date. So this is basically what we have going on in a eukaryotic or human cell, and that's what we want to be thinking about as we go through in the next few weeks and start looking at how some of these are actually functioning in the physiological sense. So thank you for listening.